We have looked closely at this fascinating and intricate process of DNA replication, a part of which involves not only the synthetic function of DNA polymerase, but also its nuclease function. In this lesson, we want to consider the latter and see how it relates to the fidelity of replication. Replication of the genome occurs once in each cell generation. Even with the specificity and function of the various components of replication, mutations resulting from errors in replication occur once in every 1 to 10 billion base pairs, and these could easily be lethal to the cell. Let's take a closer look at the proofreading ability of DNA polymerase. DNA Pol-1 enzyme possesses 3' to 5' exonuclease activity, pictured below. Without this function, errors in hydrogen bonding of incoming nucleotides to the DNA template would result in one error in every 10 to 100,000 base pairs. This proofreading and correction improves fidelity to once in 1 to 10 billion, an improvement of 100,000-fold. Recall that Pol-1 enzyme is responsible for removing the RNA primer and replacing it with DNA. It has a 5' exonuclease function that allows it to cut the RNA primer, which can then be patched by its polymerase function. Notice in the figure below that there is a single-strand nick between two adjoining Okazaki fragments as a result of this action. In carrying out this function, DNA polymerase may remove not only the RNA primer, but a few of the deoxynucleotides added by DNA polymerase III to the 3' end of the primer. The action of Pol-1 ensures replacement of all the deleted nucleotides with the correct sequence of DNA. Removal of some of the added deoxyribonucleotides results in migration or translation of the nick or gap in the backbone in the 5' to 3' direction. How very impressive are these various controls for correcting mistakes during the replication process.